Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our next talk, Applying Generative AI at the Intersection of Gene Therapy and Cancer. I'm pleased to welcome Dr. Amy Elliott. Dr. Elliott is a scientist and product leader working at the intersection of life sciences, technology development, and entrepreneurship. At FormBio, Dr. Elliott leads product strategy for biological computing, workflow development, user experience for scientific platform features, and laboratory validation of machine learning models. Dr. Elliott received her PhD from Vanderbilt University in molecular physiology and biophysics. Her scientific expertise is in genetics, light microscopy, and optical engineering and computer vision machine learning. A warm welcome to you, Dr. Elliott. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Dr. Elliott's presentation. You can also benefit from some fantastic supporting literature, which you can download for free below the video player. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. And without further ado, I will now hand over to Dr. Amy Elliott to begin her presentation.
Thanks so much, George. Thank you for your attention, and I'm really excited for the opportunity to talk to you today about the work we're doing at FormBio, working with generative AI, particularly in gene therapy, and about the exciting places that I think this may go for cancer. So without further ado, let's jump into it. I want to start with the landscape of cancer innovations as a nod to the theme of this workshop. So there are a great many innovations that are happening very quickly now in cancer therapy. I've broken them down into these three places: immunotherapies, which include CAR T-cell therapies. You'll hear a lot about those, I believe, from other talks, and immune-oncology combinations. So a couple of things to note here. There are really a large variety of therapeutics that are classified as immunotherapies, including like immune checkpoint inhibitors, which block immune checkpoints. There are monoclonal antibodies, which are immune system proteins that are designed to bind particular targets on cancer cells. There are also immune system modulators, really quite a lot of them now, which will enhance the body's immune response to cancer by either activating or inhibiting particular molecules. And so immune oncology combinations simply means combining different classes or multiple molecules in the same class of these therapeutics to boost the response. Very briefly, I guess CAR T-cell therapy is also called T-cell transfer therapy. And the idea there is you are boosting your natural ability for your T-cells to fight cancer by pulling those cells out, identifying the particular T-cells that are very active against a tumor cell, and then replacing those so that they have a better chance of fighting off the tumor. In the realm of personalized medicine, ctDNA monitoring, which is circulating tumor DNA monitoring, and single cell sequencing and spatial transcriptomics, these things sort of overlap each other. What they permit us to do is take a bunch of blood samples from a particular patient and identify with next generation sequencing what is floating around in there. If there are any tumor-specific DNA fragments or proteins or other things that will help identify a cancer, that's the realm of ctDNA monitoring. Single cell sequencing and spatial transcriptomics play a huge role, an increasingly huge role in diagnostics and also prognostics. And so a lot of these things you'll notice overlap. So in the diagnostics section, multi-cancer detection tests there at the bottom, that's also facilitated by next generation sequencing. It's also called a liquid biopsy, you might have heard it called. And so some of these innovations are really facilitated by massive boosts in technology around next generation sequencing. A lot of that is coming down to the cost, as well as just improvements in the ability to do the technique itself. And then finally, molecular imaging. This is close to my heart. Molecular imaging for magnetic resonance and PET are used to determine in vivo changes in cancer. So these mean that you're doing live imaging on a patient to see how a tumor changes its structure or morphology, which is a fancy biological word for structure. And that can give us some insight into what is happening in the evolution of a cancer. All right. Given that landscape of innovations, I want to move into AI, artificial intelligence, and Gen AI, which is short for generative AI. All of the things I just mentioned as innovations are being are strong applications for artificial intelligence. So let me talk a little bit about what this means. Artificial intelligence is basically intelligence by machines. The idea here is that we have computer models that mimic human intelligence. Very controversial whether, how and whether we're actually going to get to a general human intelligence, right? That's not really what we're focusing on here. What I want to focus on here are the applications of these tools. So machine learning, which is a subset of AI, could be defined as like the use of statistical and numerical methods that encode learning in models that are trained on a lot of data by running them. So machine learning models get better the more you run them on more data. Deep learning is yet another subset where you have multiple layers of neural networks, and what the multiple layers do is provide abstractions. And that provides quite a richer fabric, if you will, for doing a lot of different computing methods. So what I've listed here, I'm not going to walk through everything on the slide. I know this is quite a word salad, but what I did want to do was provide a landscape that you can go look up yourself if you're interested in figuring out what all of these are. I will break down just a few things here that I think are really interesting. So machine learning basically breaks down into unsupervised and supervised learning models. And what that means is in a supervised model, you have a whole lot of data that you train the model on. And then you let the model run by itself on new or test data and see what it predicts and how well it does. There are lots of computational tools for validating computationally how well a model performs, its accuracy, and things like that. Unsupervised learning is where the model teaches itself. So you do not provide training data. These are things like clustering, singular value decomposition, and principal component analysis. Excuse me. So in the realm of deep learning, really there has been an explosion of a lot of interesting and different kinds of models. These fall into the traditional unsupervised and supervised categories, but also we're seeing increasing development of some really cool things like self-supervised and semi-supervised models. So in a self-supervised model, you can imagine you give it a bunch of data and it learns itself by doing a variety of different methods, what the labels are that go with each data set. So you can imagine if I have, for example, tumor data and I have benign tumor data and malignant tumor data, what the self-supervised model will do is teach itself which one is which, right, by a variety of different mathematical computations. A semi-supervised model is one where you would provide some labels and allow the model to learn itself. So they're really in between fully unsupervised and supervised models. And it turns out there's really quite a powerful amount of broad things you can do with those models. So you can feel free to look any of these up. Some of my favorite ones are the Boltzmann machines, that's RBM and DBM for restricted and deep. And then variational autoencoders, VAE under semi-supervised. I just think those are super cool. But basically, like in the realm of diagnosis, and also in treatment and in prognosis, you're seeing an increasing amount of these models being utilized as tools to make that faster and more effective. Obviously, this is important. There still has to be a human in the loop. So we're not at the point yet where any of these things are really performant enough to replace a physician or a radiologist. But what they are doing is really helping shorten the time to the best of diagnosis, treatment, and prognosis. So what I want to do now is take a little bit of a deeper look into generative AI models. So natural language processing and computer vision sort of span both, all of the above of AI, ML, and DL. Generative models are machine learning models that learn the patterns and structure of their input training data. And then they generate new data that has similar characteristics, right? So it's kind of self-explanatory by its name. So I want to focus on the large language models, which are getting a lot of attention right now with ChatGPT, et cetera. and GANs, which are generative adversarial networks. So you'll notice on the slide that these fall into self-supervised and supervised categories. However, that's a little bit challenging because they are made up of, a lot of them are increasingly made up of more than one type of model architecture. So briefly, large language models operate on text data. They are composed of transformers, which was a huge innovation in machine learning in about 2017, 2018, something like that. Maybe the first one was a couple years earlier. And they have self-attention, feed-forward, and normalization layers. So remember that deep learning is composed of neural networks. Neural networks have multiple layers in deep learning models. Applications in oncology for these large language models include things like extracting data from electronic health records to more quickly do pattern matching, drug target discovery, and phenotype prediction from gene expression. These ones are really interesting because if you think about it, the nucleotide code, the amino acid protein code, those are all letters, right? Genes can be thought of as words in a language. So applying the large language models to the language of life, if you will, I believe Francis Collins has called it that, is a really, really interesting and powerful tool. And then lastly, protein design, again, because proteins are made up of amino acids, which are letters. And computer vision, this is another really big area for generative AI in oncology. The idea here is they train on image data. They have two components, a generator and a discriminator. And so applications here are classifying benign versus malignant tumors from pathology slides. Radiotherapy is an increasingly interesting use case for GANs. They can be used both in diagnosis and planning. So, you know, identifying like where exactly it is that you want to apply the radiotherapy, what the dosage needs to be, things like that. And lastly, in screening. So when you're looking at MR images or PET scans, these are useful models for helping make that, you know, time to determination faster. So what we're doing at FormBio is focusing in on the natural language processing models for the moment. So we'll focus more on that. I want to talk a little bit about all of these, what all of these models in the context of challenges and opportunities, what all they provide. So in the space of cancer, I feel like challenges and opportunities are sort of tied together. There are many challenges in every step of dealing with a cancer diagnosis. Some of the ones that lend themselves to the promise of AI and machine learning and deep learning techniques include early diagnostics, that's radiology for image processing, screening for multi-cancer detection. There are opportunities and challenges in efficacy, right? Not every drug is going to work for every patient, right? And so outcomes are a particularly critical component. So sequencing technology and the tumor microenvironment, which we're starting to pay a lot of attention to because we can do things like spatial transcriptomics, which give us a really good picture of what's happening in tumor and microenvironment. These are places that are particularly susceptible also to opportunities in AI, but they are also challenging. Economics is going to be forever with us as a persistent challenge. Drug discovery, regulatory approval, monitoring during both clinical trials and therapeutics, and just the general bloated cost of healthcare are real economic challenges. Some of these things can be sped up, right, by using these sort of machine learning techniques. And I think that is the big opportunity here, is to bring down that cost. Lastly, manufacturing. Manufacturing is a challenge, both in terms of protocol iteration, but mostly in terms of time. Therapeutic optimization, getting enough doses per manufacturing run. These are also challenges that have been only, I think, recently super appreciated because the FDA is starting to make recommendations about what the outcome of a manufacturing run should be with respect to gene therapy in particular. So that's where FormBio is really focusing, is on the biomanufacturing for therapeutics. All right. So just to give a little bit of an overview of what that looks like, as you develop a drug from the discovery, safety, and efficacy phase, let's bring this into cell and gene therapy in particular. So this could include your CAR T cell therapy, where you're pulling a cell out, modifying it, and then putting it back so that that T cell is more effective for hitting up that tumor. So when you get through the discovery, safety, and efficacy phase, all of that is in vitro. It's at the cellular level. Then you have safety, toxicity, and efficacy phase. That's in vivo. And then you get into trial preparation, manufacturing, and FDA checkpoints. So during those first two phases, discovery, safety, toxicity phases, you're basically doing small bench level scales of manufacturing your product. Scaling that up so that you can create enough material for your clinical trials and your actual dosing of patients is a huge burden on the manufacturing facility for optimizing to get the right dose. So this is just a little bit more detail on that, but basically some of the major things you have to worry about are low yields, especially for cell and gene therapy. I'll get into a little more detail on that directly. Low yields is an issue. Safety issues are a real problem because you end up with empty adenoviral capsids, in particular if you're using a viral vector for delivery. That has all kinds of immunogenicity worries. The iteration on biomanufacturing, which is the big thing that we're focused on, and then of course getting through approvals is getting increasingly challenging because we are upholding very high standards for FDA approval. So the idea here for our purposes is, for FormBio's purposes, there are the opportunity to reduce the cost to the company who is developing the drugs, which is big because we want them to keep developing these drugs, right? So we can save millions in the biomanufacturing cost. We can reduce the elevated risk of immune responses if we optimize for biomanufacturing. And then in terms of the patient cost, which is both physical and economic, we have overlap at this elevated risk of adverse immune responses. We really want to bring that down for both the therapeutic developer and the patient. And then the prohibitively high cost per dose is often felt by the payer and the patient. All right. So what are we doing? Our AI/ML/DL architecture, really, it's all of these things. We have layers of all of these things, right? So we have our own optimized processing and delivery engine. We get about 18 times faster output, 5x cost effectiveness, and a really ridiculously huge reduction in memory. It's a cloud native architecture. We have things like a transformer neural network. This goes back to that large language model that I talked about earlier that we're applying to DNA and protein analysis. We're doing coding, training, and inference on pre-trained models for things like methylation, variant effect prediction. That's where all the sequencing data becomes really important. And then what we're doing is using that framework for machine learning models that identify truncation propensities. What I mean by that is when you're creating a gene therapeutic product, you have a gene that you have to get into a viral vector if you're doing a viral delivery system. And then you've got to get, so that packaging process of getting your gene of interest into the viral capsid is fraught with all kinds of issues. And one of them is that the product ends up truncated and you don't have a full therapeutic product in the capsid. That leads to the immunogenic response and other things. So what we're doing is identifying those truncation propensities, including like the diversity of them and the magnitude. We're doing this with a variety of data sources, including a lot of public data. This has been an incredible push by funding agencies and also just academics to make their data public for use by others, and also a lot of data that we're collecting with our customers and clients. So here's just a graphical image of the idea that you've got to get your gene of interest, your transgene, into a capsid, which requires co-transfection with a helper plasmid, and usually also a packaging plasmid. So it's typically a triple transfection. You dump all that into a cell, and some percentage of those capsids will be filled with the thing you care about. There are about 29 FDA-approved gene therapeutics right now. There are more than 100 of them currently in clinical trials. So this is a huge opportunity space for improvement and the use of AI for that. So what we're calling our product is FormSight. It's composed of basically 3 steps. We've got a bioreactor simulator, which simulates the cellular environment for packaging of that DNA gene therapy payload or product. And then we've got an optimization step where we are optimizing that product to reduce truncations and produce a larger number of doses per manufacturing run. And then we can check our optimization to find out how that's going to perform in the bioreactor before we ever get to a bench. So that's the big thing that we're working on. So Formsite Simulate, basically we take in your gene therapy construct, we take in your packaging and your helper virus, and we have a series of rate equations and models that simulate the cell. And so what we're able to do here is basically see how the manufacturability of those parameters will scale. So we could test multiple bioreactor volumes, for example. Mostly we're looking at suspension cells right now, but we're also looking for opportunities to spin that out to other models. All right. This is an example report of what our Formsite Simulate produces. And so some of the really cool things that we're able to predict with this is the full capsid titer. So we're getting the vector genome titer. We have the rate of filling. That's this graph on the left here. So we can see, when we start getting a whole bunch of capsids full of stuff that's not therapeutic. We can predict the percentage of full capsids, truncated products, and chimerics. That's going to be a mix of constructs that have backbone and maybe some of your gene product, or backbone and packaging. Like, there are a whole bunch of different combinations that make up chimeric. And we're predicting these based on a ton of sequencing data that we have. Truncation distribution by region. So we can tell you on the truncated, products where they came from. Is it the promoter? Is it the coding region itself? And then we can look at, what the secondary and tertiary structures are, whether they correlate with where those truncations are. And that's this graph on the bottom. So this gives us a lot of information about where to focus our attention for this optimization step. We can swap out promoters. This is another place where really matters what the regulatory landscape is. You can't just willy-nilly, swap out components and expect very new things to go through FDA approval without extensive testing. So what we're doing right now is swapping one approved promoter for another if we get better performance. And then our machine learning tools, our AI tools, will optimize the coding region to reduce those truncations. So basically what we did here, and this is where the machine learning becomes really useful, is we had a bunch of data from several different constructs. We looked through it to identify structures that correlated with high regions, hotspots of truncation, and we use those features to train our model that will predict where it is that we expect to see those truncations. And then we can do a codon substitution to keep the same amino acids for that coding region, but that gives us the opportunity to change the sequence to improve the manufacturability. And that's really the thing that's exciting for us to do, is how much better can we make the manufacturability in these constructs by applying our deep learning models. So what we're able to do on some of our examples, we can increase our full reads, that's sequencing jargon, so we can get more therapeutic product by 18%, and we can decrease the truncations by really quite a large amount for some of our constructs. And then we simulate again and see how we perform in the bioreactor. And in the best case scenario, these don't look all that different, but really it's a 4x increase in vector titer. And if you look at the y-axis, it's really an incredible improvement that we're able to simulate. So most important part of this whole thing is, now that we've got all of this AI and ML and smart things happening on our manufacturability question, right. So sorry, this is just the benefit of getting this all working correctly. Is we reduce the cost, we reduce the time to market, and we, more importantly, get more patients treated per biomanufacturing run because we're able to produce more doses. This is the important part. The important part is, now that we have this framework worked out, the thing that we want to do is keep pushing the field forward. And I mean that in both computational land and physiology and biology land. So we are analyzing a ton of NGS data, next generation sequencing data, to understand the heterogeneity we get, that chimeric versus truncated versus full capsids. We're resequencing a bunch of our constructs that we use for training to test the fidelity of our training data. And we're producing optimized and controlled products at the bench scale to validate the predictions of our machine learning model. And this is the part I'm particularly proud of. So for resequencing, what we're seeing is that, a lot of sequencing technology, I said in the beginning, has changed very quickly, right? So the 4 or 5 years between the first set of sequencing data that we trained our models on and the sequencing data that we're using today to validate or check the fidelity of that training data, they're completely different machines. There's been a hardware upgrade, a firmware upgrade, the software and the processing are pretty different. And so I feel like an important question of like, how well does our model hold up when we're using data from these much more recent, better, technologies? And really the answer is pretty well. So our truncation distribution predictions are pretty similar. We see a consistent forward and reverse asymmetry, which is really pretty expected because 5' to 3' and 3' to 5' are not the same. And the sequencing read distributions themselves are very similar. So the magnitude is different for what our proportions are predicting, but the distributions are very similar. So that's helpful for understanding how robust our models are with respect to the data that we trained on. All right. Lastly, for producing the bench work, the bench scale validation, we are really, really excited about. So this work is underway. I will hopefully have something more exciting to present on that in the future, but we're basically running all of our constructs through the whole packaging process. We're collecting vector genome titers. We're getting the measures that we need for whether or not we produce the correct protein, things like that, so that we can, and we're doing next generation sequencing on all of that so that we can really validate and make sure that all of the in silico predictions that we're making on our models really mean something in an actual cellular biological landscape. So who the heck are we? Well, FormBio is a company that provides software and, like I just spent a lot of time talking about, machine learning-based solutions with the goal of empowering cell and gene therapy developers to address the biomanufacturing challenge, which is a pretty big one. So we have a whole platform that we perform all this compute on. It's a native cloud architecture. We have a lot of data management tools, workflow execution. So we do a lot of our own next generation sequencing processing, for example, with our own workflows on our own platform. We were founded just last year. So really it's been quite a whirlwind over the last year, but we've got a lot of really exciting things happening in genetics research and cell and gene therapy. We've got a whole suite of CRISPR tools for synthetic biology, which is another place that I feel like is really up and coming for cancer in particular, is the use of CRISPR for CAR T cell therapies and other things. So we're really excited about the potential intersection of all of these tools that we're building with cancer. So again, just core areas where we are operating our bioinformatics platform. We're also adding image processing, so that gets us a little more into computational life sciences broadly and less strictly in multi-omics. Cell and gene therapy development, we've got a whole incredible roadmap that I'm really excited about for that. And then gene editing and genetics, that's where a lot of our CRISPR tool sets come in. We were spun out of a company called Colossal Biosciences, which is the de-extinction company. And a lot of the gene editing and genetics research tools that we built were built in collaboration with Colossal. All right, so where are we going from here? The future landscape of cancer innovations is really going to include, I think, a lot of new tools and exciting things in data exploration and analysis, protein engineering. That's good. That's getting a lot of attention right now, and I think it'll be really exciting to see where that goes in the next couple of years. Same with precision medicine. With all the new sequencing techniques, that's going to be a really incredible place to watch, I think. And then design for manufacturing, which is where FormBio lives right now. Goodness, there's a lot of exciting things happening, and generative AI is really pushing a lot of advances in those spaces. With that, I would love to acknowledge my amazing machine learning team and our bioinformatics team at FormBio and the engineering team that has really worked very hard to put together a platform that we can do all this amazing work on. We have some really incredible collaborators, both in industry and academia. I want to mention the UT Southwestern Medical Center, which is collaborating with us on a lot of our bench validation. Google, PacBio, and NVIDIA have been incredible partners for multi-omics, for our next generation sequencing analysis, and really accelerating our compute so that we can do these things extremely efficiently and very quickly on the cloud. Thanks so much for your attention.
Thank you very much, Amy. That was a fantastic and insightful take on generative AI. As with all the talks, we are likely going to have lots of questions from the audience, and I can see we have received some questions already, so I'll move on to your first question. What is the current status of manufacturing and drug development and the top focus areas for change?
Thanks for that. That's a great question. So it sort of breaks down depending on whether you're talking, what kind of molecule you're talking about manufacturing. So there are different challenges in small molecule versus cell and gene therapy manufacturability. And so some of the main focuses and top issues are on, capsid design and vector design for manufacturing gene therapies that are going to have less immunotoxicity, that are going to have a better tropism for the right tissue that you want to get it to. And the other thing that's going to be a common problem and focus is the time to effective protocol for manufacturing and the and getting a high enough yield to make it worth the incredible expense of all that iteration to get a particular gene therapy product made.
Thank you, Amy. Moving on to your next question, can you speak to any case studies at this time that clearly show the AI's potential to impact the CGT company's economics, market timing, and ability to impact human health?
Goodness. So there are some, we have ourselves some case studies that will probably publish in the next few months, where we're doing experiments in collaboration with a combination of gene therapy developers and manufacturing facilities and academics. In terms of case studies that currently exist, there are a few publications that have come out recently, and I will have I think in the next month, there will be a blog post on FormBio's website where I will have all of the references from this talk, and that does include some of them. So I'm happy to provide that, on our website later next month.
The next question I have for you is, with the announcement of FormSight AI at ASGCT, how do you think this solution can shift manufacturing processes in preclinical cell and gene therapy development in the next 5 years?
Yeah, that's a good question. So I think the most important thing that we're looking at changing is the time to manufacturing protocol that's going to be enhanced. So I think there are a few opportunities here that we are really excited about. One is anybody that is doing bench work for even in small scale for making virus particles that you're going to infect a tissue with, getting the right transfection protocol, getting the right purification protocol. There are so many steps that have so many different parameters that are challenging for optimizing. So you end up iterating. Some companies are telling us that it's 9 to 12 months for a particular therapeutic to figure out the right mechanisms for manufacturing, for packaging, viral packaging, and then for purification downstream, right? So that is a lot of time, a lot of money spent just figuring out what the protocol is going to be. When you're a grad student, it's like, that's part of your life. But when you're talking about therapeutics, that becomes a whole different issue. And so we're really excited about bringing that time down. Our FormSight platform is extensible also beyond just what we're looking at right now, which are DNA-based therapeutics. So we're also really excited in the next 5 years about moving into other viral delivery systems like lentivirus and other payloads, like, for example, shRNA or mRNA or miRNA type delivery mechanism.
Thank you so much, Amy. Moving on to your next question, how can generative AI techniques such as GANS, VAES be employed to enhance the development and optimization of gene therapies for cancer treatments?
Sure. Yeah. So, couple of things here. So, GANs are that's mostly GAN is mostly used in computer vision, and so there are lots of really cool things that you can do with. For example, let's take a histology section where we're looking at tumor material versus benign wild type normal material. You can train a GAN on the wild type on a subset of wild type data, and you can feed it a bunch of tumor data and it will recognize what the differences are in between those two images. So you can generate a lot of images that will help refine that model. And the thing is, right, you have to keep a human in the loop for these things because they're prone to artifacts in some respects. However, the excitement is it reduces the number of people that have to look at it, and it reduces the time to identifying what the features are that define a tumor versus a non-tumor type tissue. And so the hope there is that it will make diagnosis faster, and it will make it will help us learn what we need to go back to the lab and figure out might be real features that perhaps push the field forward for earlier diagnosis. And then for VAEs, so the autoencoders, those are really, really interesting for looking at things like the variation in a sample set. So if we're looking at gene therapy is going to, it's going to have a differential impact in a gene that is maybe you've got a haploinsufficiency, for example, but it is not 100% penetrant. So you're going to have a range of a population of patients that have a sort of different level of clinical impact for whatever their genetic disease is. And so, you can imagine using something like a VAE to understand what the variability is in that population and help figure out like what the likely prognosis is going to be for a therapeutic or how effective that particular treatment is going to be. And so those are things that are sort of really in early stages of exploration, but possibly really exciting future states.
Thank you, Amy. We have a couple more questions for you here. What role can generative AI play in predicting the efficacy and safety of gene therapies before conducting clinical trials or experiments?
Yep, that's a really good question. That's an area that we are starting to look into ourselves that I'm really excited about, which is, so a little bit of background on particular viral gene therapy. There are quite a number of vectors that were derived from an original adenovirus, right? Adenovirus has a lot of triggers for an immune response. It's a fairly immunotoxic virus, and so the number of sequences, the amount of adenovirus sourced tissue sequence that ends up in those vectors is immunotoxic. And so to the extent that you can figure out what those are going to be and reduce them with an optimization protocol, that's a place where I think generative AI may play a really useful role in identify at least for what we're looking at for the large language models here at Form. So we can identify the sequences that are likely to be, for example, there's a whole lot of literature on CpG islands and GC content driving toll-like receptor 9 immune responses and things like that. So the generative AI has a real opportunity for anything that's going to be a potential nucleotide trigger for figuring out where in a given vector we are going to have that problem, and then hopefully figuring out how to replace and change that around to make it less immunotoxic before going to clinical trials. Yeah.
Thank you. Moving on to your next question, what makes FormBio's platform unique compared to other AR cancer research platforms?
Yeah, sure. I love that question. So, so one of the main things that we've got is a really incredible team of experts. We have biologists, we have people with PhDs in oh my goodness, I'm blanking on whatever your immune field is called. Right. We've got a bunch of people that have physics and AI backgrounds. We've got a lot of really strong computation, bioinformatics, and multi-omics expertise. So I think a lot of one of our main advantages is our people. We've got a really cloud optimized platform. So we've got data provenance and like all of these things that the cloud provides in general on our sort of underlying back end. And then I think the other thing that's really important is that all of the machine learning and the really accelerated bioinformatics tools that we're building, we're testing. So we're working with lab partners to make sure that everything that we build in silico has a real world impact in vitro and in vivo.
Thank you. It looks like we have some more time to answer a couple more questions. What hesitations towards the use of generative AI have you most experienced within the industry, and how do you respond to alleviate these concerns?
That is a great question. So, as a scientist, I have to exist with a certain amount of skepticism, right? A certain amount of healthy skepticism, and I think there is a lot of concern and slow adoption for AI, which is I think appropriate. So I think the main thing is going to be what we're hearing is that the main thing is going to be the bench validation. So being able to demonstrate that the predictions that you make with your machine learning models actually, come to pass in the lab is going to be, I think, the main differentiator for assuaging these sort of fears and easing the adoption rate. So I do not think that the right thing to do is validate a subset of machine learning models, in particular the large language models that are getting a lot of attention right now, and then saying, hey, this one worked, let's do this with all of the rest of them, right? I do feel like it's important to maintain that each of these tools works best in the context in which it was developed. We do not have anything like general AI yet, right? There is a lot of discussion around whether these things are cognitive and thinking and aware and whatever. And we're really a long way from being confident that any tool that we build that's generative is going to be actually accurate. It's going to have a diminished bias, and it's going to produce results that are really reproducible. And that is, I think, the thing that we need to spend a lot of time on before broad adoption. Everything should be evaluated in its context, and it should be evaluated very rigorously.
Thank you, Amy. I do have one final question for you. What timeline would you estimate for adoption of automation and infrastructure tools in life sciences?
Oh, that's an interesting one. So, so it's already happening in a few places. So there are a few places where, for example, you're seeing robotics in the laboratory. So I've seen at least one really cool lab for fruit fly research. So fruit flies grow in little vials of food, right? And when they go through all the food, they have to get flipped over into a new vial. So you're seeing robotics for flipping fly vials. You're seeing robotics for moving samples from one machine to another for sometimes purification steps and things like that. So I think we're starting to get to an age where automation is a reasonable expectation if you can afford it. So we're only seeing that right in places that have lots of money because it's expensive. And then I think the next thing beyond that in the next few years that we'll see more of broadly is the marriage of the robotic automation with cloud infrastructure for direct data transfer to platforms like FormBio, where then you can do downstream analysis, you can explore the data, you can do there are a lot of really interesting things I think you can do. So there is an emerging concept of sort of a digital contract manufacturer, for example, where you can track a sample that comes from the very first, ingestion from a therapy developer and goes through the entire manufacturing process, totally digitally tracked, exactly what happened at every step of that sample. And I think once we get enough of that data, that'll also really power the next generation in the next 5 years of machine learning models for understanding advanced manufacturing and getting the right steps for purification and all these things.
Thank you so much, Amy. Unfortunately, that's all we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Dr. Amy Elliott, and we'll be back soon with the next talk.
Thanks so much.